So our next talk now is gonna be myself and Manjula Senanaik. We're gonna give a talk on some preparation and experiment planning. Let me share my screen. I hope everyone can see. So the talk, like I say, is gonna be on some preparation and experiment planning. Important stepping in Sun's bean time. So is a very important to have the right sample and the right conditions to be ready for the bean time. So going over some critical points that is important for some preparation. So some preparation is a very labor intense work before the bean time, and even during the even during the bean time, we can start to say that during the expression or the chemical synthesis of our biological sample, doesn't need to be only proteins or nucleic acids, could be biomaterials, lipids as well. So in that point, we need to define what is the contrast match point we want to target. So we need to define which SLDs of the buffer or how much D2O in the solvent we need to have so we can properly contrast match our target samples. And also we want target high contrast conditions in the way that we can reduce the incoherent scattering and increase the coherent scattering. So purification is also a important step. I'm not gonna over all those topics, but there's many ways where we can get pure samples during some preparation. During the presentation, we will see some of them. And it's very important to have a preliminary characterization before the bean time. So using a gel filtration or size exclusion chromatography, dynamic light scattering, small angle X-ray scattering. So you can observe if our sample it's monodispersed or it's aggregated. Also detecting the particle interference, which is going to cause an issue during the bean time. So we want to avoid that as much we can. And also found out if our sample has some heterogeneities that you need to know so you can properly analyze our data after the bean time. And also, which is important, know if our biological sample is happy in D2O. So some characterization using those techniques can really give you valuable information for you uh, to uh, to provide a good sample for our experiment. So sample preparation prep in time, you can use some device to optimize the concentrations that we're going to use. So you it's better have a good know before the meantime what protein concentration or sample concentration you need to use to give you a good data. Separate multiple species as much you can. So if you have a mixed population in your sample, you have to use some kind of column system that can allow you to separate those species. And also, it's very important here is where it can give you a big trouble if you don't have the correct SLD or correct percentage of D2O in your solvent in order to do the contrast match or the contrast variation series that you are doing right. So volumes are critical during some preparation. We need a kind of a larger volume, 320 microliters of sample to do it's not large compared with the other techniques, but it's still a lot of materials to do a SUNS experiment. So it's better have enough material so you can prepare all the samples. So concentration should be already optimized before the bean time, so you can target where or how long you're gonna need to measure those samples. Like I said, the duration level in the schedule range dense of the sample components, and the buffer should know a priori. Uh, and also is necessary for student proteins and nucleic acids and lipids. And lipids is not the case, but you need to have monodispersed species in order to have a proper way to analyze the data. Yeah. Here you go. So when I say that we need to have monodisperse samples for solution sketching, what I mean is that our sample should be well behaving. Here I'm talking about homogeneous particle like those fishes in solution where I can distinguish the scattering for each particle in my system. So from this monodisperse profile here, which is the reduced data, the average reduced data here, I can extract the information about the part distance distribution and get the max from this data here. The overall size, which is represented by the rates of gyration, and other informations as well. And I even do comparison with atomic structures or high resolution structures and get a comparison with models that can supply my scattering profile. In this case here, we are in monodisperse condition, and then we don't have the structure factor, which is close to 1 for a dilute condition. So in my slides, I put some experimental conditions here, protein concentration and D2O. We will share the slides with you, if you will, after the workshop, so you can check what was the concentration and the contrast conditions that I use for those samples that are present here. Preparation of monodisperse samples. How we can prepare monodisperse samples? The preparation starts, uh, in this case, inject your sample in a size exclusion chromatography. Doing those kinds of purification, you're gonna help you remove large aggregates of our samples, which is gonna be separate here in the beginning of the dilution of the size exclusion. It's one thing that it's important to know is that those large aggregates they not show large absorbance in the UV spectra, but they have a very power to do light scattering. So you may not see then the large size exclusion here, but they will show up has uh, large species in your scattering profile. And the page analysis, which can give you the idea of the molecular weight, and also the if you have contaminates, you should observe a single bond there. And in this case, using centrifugation can help you a lot during the bin time because uh, if those aggregates can be removed using centrifugation, you can save a couple hours of bin time. For instance, you can easily lose 12 hours of bin time if you don't properly centrifugate your samples before the experiment. So in that case, if everything behaves well in this step here, you have high purity samples and you have a good chance to have a monodisperse species for bean time. Uh, so here is one example, one science example where the initial characterization helped us to define what was the ideal sample for the SUNS experiments. We have a single band on the page analysis, and we have a single peak on the size exclusion chromatography. Small species was separate here. But when you did the SACs using different protein concentrations, what we observed there is still have the presence of large aggregates on the sample. So as we increase the concentration, those large aggregates are even show up more strongly in the low Q. So this kind of sample can corrupt our data and not allow us to do the proper analysis. In this case here, the main issue, we have the sample in good buffer, good salt conditions, but the his tag, which was used to purify this protein here, was causing the aggregation. So we removed the his tag of the sample. We thought that we could leave the his tag there, but this was not the case. And we removed the his tag, and then we got monodisperse profiles, having go to 8.5 milligrams per mole, which is a very high concentration. And here we see the SUNS profile uh, of the same sample we collected here. And that is the X-rays data, and here is the SUNS data of the same protein that we analyze. This sample was deuterated. We express and deuterated in E. coli, and then we did all steps of purification, going through the centrifugation, and until get the SUNS experiments, which was the last step. So ORNL offers the opportunity for users to do the initial characterization of their samples. We have a success instrument that Kevin showed for us on his tour. So we can collect solution samples here on Warsax machine. We have a 96 plate well here, and the flow cell can load up to 100 microliters of sample. The target concentrations in general, from my experience that I have here, for instance, you have a 10 kilodalton sample, you need to get good data with good signal to noise around 5 to 10 milligrams per ml. If you increase your molecular weight, your concentration can be decreased to 2.5 to 5 milligrams per ml. And how long we're going to collect our data is going to define this statistic. So for those samples, probably 2 hours, 3 hours collecting data for sample and buffer is going to give you good quality data. If you increase the molecular weight, then your concentration can be even lower. So, and those concentrations is kind of close for what we can measure in the SUNS as well. And if you want to get more data with high quality and high resolution, we can use our JoinSS program with NSLS2 SL2 to collect a sample at high output stacks and also do sex stacks, which is mean that we can do the purification, use size exclusion, combined with stacks. If you want to have more information about this program, I'm happy to give the guidance how we can assess the Sachs Binline in combination with the SUNS proposal as well. So here is some issues that I want to highlight, that when you have samples with impurities or mixed populations there, how those affect our data and the way that they manifest. If you have a low molecular weight species, probably, for instance, here we have 2% of which is low molecular weight species, not gonna be causing interference too much in our data, because our dominant scattering species is our target protein or target molecule, and does not affect too much our P of R profiling, for instance. But if you have a high molecular weight species, if it's only 2% of the total mass you have there, and your sample is the molecular weight is lower, this molecular species contaminates our data, corrupts our data, and dominates the low Q. So you can see this behave of the P of R getting a very long tail here on the large distance. It's because of this mixed populations where we have only 2% of a large molecular weight species contaminating our data. And if you have here, which is in the last case, a pure sample is only one single bundle on the page analysis, but the scattering profile does not agree for only one conformation. In that case, in this case here, we have a mixed populations of different conformers that is gonna give you a different P of R profile that we should expect for a more globular structure. And that case can be separated computationally, or you can use the size exclusion to try eliminate those conformations. But in general, those conformations are transient between one each other, so it's very hard to separate them. This case here is a SUNS case of protein, which is it was not homogeneous, so it's a heterogeneous samples. So the blue line here in the size exclusion chromatograph, as you see, there is a very long tail. Is not the size exclusion does not have a bell shape. Every the sample are pure. So we have here is a it's a protein complex. We have two proteins in your systems, so they form a kind of transient complex. So you have monomers, dimers, and the tetramers of this protein here. And the way to analyze this data, the sample is still monodispersed, but then you need some kind of computational tools to help you separate those conformations, and even using other techniques to help analyze those kind of data. So this transient behavior also is transferred to when protein interacts with DNA or RNA, for instance, we have this transient complex here collapsing for two subunits, which is the NSP7 here and NSP RNA. So these kind of samples we can analyze, but then we need to know a priori the characterization, how the sample behave in the size exclusion, and what kind of information we can start from there to know that the sample is not only one type of particle. There are multiple species there. So like I said, the centrifugation can really help remove aggregates. In this case here, how we will see that our data is corrupted during the bin time. In the first line here, in the first column, sorry, we have a good data set where we have a monodispersus piece. We can easily fit a Guinea profile. The Guinea is linear across a wide Q range. So our P of R is well behaved, and we can extend it to the maximum, and the P of R does not oscillate in the X here. So that tells us that the sample is well behaved as well. The monospace of the sample it's good enough for you to get good quality data. And when you start having interparticle interference, here is the case where we have a repulsion. Is not very strong repulsion in this case, but subtle repulsion, where you see downturn in the low Q. So this downturn, it means that the sample does not like to be close on each other. They repel each other very strongly. And what you observe in the P of R is that you have the D maximum go very shortly, uh, approach very shortly the maximum distance. And you see the difference between the regular PFR, which is monodispersed, to go to a D maximum, which is go too fast to the D maximum. And if you extend the word D maximum, you see a negative PFR here for long distance. That tells you that our data was corrupted. In the last case, we're having you see a upturn in the low Q, and then where D maximus is going to be going to the maximum very, very, very fast in the for large distance. And if you extend where the maximum, till the maximum start oscillating in the positive axis for higher distance. So that defines some kind of issues that you can have in your data that can be problematic to from the structural information that I show you before. Um, Types of the interparticles interference that we can easily detect in one sun's experiments. So this sample here was it's a 9.2 kilodalton protein. We inject on the size exclusion, we see a single peak here. Everything looks fine. This sample here was performed size exclusion chromatography before the bin time, and you see here the presence of some aggregates, but when you prepare the sample, the time you prepare the sample and bring to the bin was enough for the protein fully aggregate, which is the green curve here. So we have a very high aggregation of the sample when we stood the sample. But which we could solve this issue just doing a size exclusion chromatography sense that Kevin told was, which he is leading the development. And then just after the bin time, just after the purification, we could collect a monodispersed sample. So that is an aggregation which is time independent and have several impacting our data. In this case here on the right side, we have different kind of interaction where we have the repulsion, and we have here the strong structure factor that shows up in the high Q. So the way to reduce it is interactions. The way to solve this case here was doing reducing the concentration of our sample in A-fold in order to eliminate this structure factor and eliminate the interparticle interference in the low Q. So you see here in the Guinea we have a linear regime where we have the downturn of the low Q. So the deuterated strategy for SUNS with contrast matching, in some cases, you may don't need to use deuteration if you are studying protein, DNA, RNA complex, or in some cases lipids. They have the moiets of those molecules. They have different contrast match points. So for in general, for always for a proteated samples, the contrast match is going to be 42% of D2O. And for DNA, RNA is around 65, 70% of D2O, and a lipid around 10% of D2O. So knowing these contrast match points, you can design what is going to be your son's experiments. And if you need deuteration, you can also target what level of deuteration you needed. Please talk to us. We have a good experience to know what are the level of deuteration for bunch of different systems that we can already apply to our system, so we can target the deuteration you need. So in this case here, we have a protein lipid where we want to contrast match the protein in we do the contrast match where we have it, sorry, we have here a nanodisk where we have the membrane protein stabilizing this nanodisk. We want to contrast match the nanodisks. In this case here, we have two proteins, which is scaffold proteins, which allow us to form these nanodisks and stabilize the membrane protein. In this case, we cannot really solve the problem because we have two contrast matching, which is identical for the protease samples. And the lipids are contrast match and low percentage of D2O. So if you contrast match lipids, you should see the two membrane scaffold proteins. By using a combination of the partially deuterated DMPC, which is the lipid used in this case, and proteated DMPC, we can raise the contrast and also create a condition where we can contrast match out the lipid. In this case here, the way that we applied this was to deuterate the membrane scaffold. So we matched the contrast match point of the lipids with the membrane scaffold and keeping the membrane protein stabilized proteated. So the next strategy that we use to do protein deuteration is like Kevin told in his presentation. In general, we express the protein in some cells, E. coli, to produce a deuterated protein. And then the next step is going to be doing the contrast variation experiment. So we do a contrast series. So be prepared to do a proper contrast series with at least 5 samples, 5 contrast points, where we can determine the contrast match point of our deuterated sample. And you can use those proteins. In this case here, we are co-purify the NSP7, which and in complex with NSPA. So NSPA was proteated. Yeah. Sorry. And NSP7 was deuterated. In this case here, the formation of the complex is a partially deuterated complex. And then you can go to the contrast match point of the complex, contrast match point of the deuterated sample and observe the scattering of the proteated one. So we can separate, that way we can plan and separate in advance what target you want to study. So in all of those processes that I'm talking here, one that is very important is the buffer exchange to D2O. So I usually do 2 different ways here in the lab. One is dialysis, where you do a series of dialysis to remove the proteated buffer of our samples exchange, for instance, here for 100% of D2O, or whatever buffer condition you want to use. And then by the end, you have a equilibrate buffer and sample in the same D2O content that you can use. So this solvent in the buffer here is going to be our background for the subtraction during the some the bin time. And also you can do concentrations, a series of concentrations and dilutions where you concentrate your sample down and uh, you load more both the buffer you want to do the final concentration of the D2O you want to use, and you do this step multiple times until you get a full deuterated buffer and the level that you want to do. So it's very important you get the right D2O in your buffer for the experiments, like I said, because it's going to define your contrast match points, the SLD to do the contrast match points. So a few another tips for preparation of the bean time. Know very well the protein concentration or whatever concentration you're using for experiments, and take very good notes on when you are doing your bean time in your system, so you can know where you are. So it can also help you doing data analysis if you know those numbers very well. So you can use this. You can use UV is using the absorbance in 280 to determine the protein concentration. But if your protein does not have absorbance there, you can use Bradford assay or similar to determine your concentration if your protein lack aromatic residues. In this case here, those for protein aggregate is good to know. You characterize where this aggregation comes from. Sometimes the system you aggregates, and that is the nature, is the thermodynamics of the system that causes aggregation. You cannot really avoid that, but it's very important you to know what was the source of aggregation and how you cannot maybe avoid that. Like I say, how our samples behave in D2O mixtures. So sometimes if you do the concentrations and dilution series where sample does like to be at a higher concentration when you are doing dilution, so the best way to do the buffer exchange is using dialysis. So that is kind of tricky. It's good to know way pure the bean time, because when you are here, it's so many things that we have to go over. And if you have those issues in the day of the bean time, maybe it's going to be critical for war experiments. And some groups also does the buffer exchange during size exclusion chromatography, where the buffer where they using to do dilution or purification is in D2O. So it's another uh, patient as well. So we were requirements for SUNS is around 220 microliters of sample for one measurement. So with that we can you can make one sample from, you can measure your sample using 320 microliters. So protease samples must target high percentage of D2O in the buffer, so you reduce your background, and high protein concentration, the better. So here I put a good numbers, some numbers that I thought that uh, is in the range that I have been seeing, that we have collect good data for those concentrations here. But each case is a case, so please talk to us, and then you can help you design your son's experiments. Now I'm gonna hand to Manju, which is gonna give you the talk about hierarchical systems. Manju. You're muted.
How about now? Can you hear me?
Yes.
Okay, thank you. So thank you very much, Wellington. So as you promised me, you exactly finish at on time. So I have 10 minutes to complete my 7 slides. I think I'm going to finish it before the 10 minutes because I strongly believe I can do that because I'm born in raised in Sri Lanka, which is closer to India, so I can speak very fast. So in these next few slides, I'm going to talk about how hierarchical systems going to be measured at SANS beamline and what are the practical challenges. So I'm going to show you some of the practical real life examples, but I'm not going to focus on the real science. But if any one of you are interested about the real science, we can talk in the offline. I'm going to really talk to you what are the challenges, how we make the samples. So basically there are three kind of samples are coming to measured at the bioscience. One is the homogeneous sample, which is covered by Wellington right now. Basically, those are proteins in buffer, DNA, and RNA in solutions or virus particles. It's very hard to make the samples ready for the beamline, but after the samples are ready, it's not very hard to fill into the cells. Like usually they are using the Banjo cells to fill the samples and do the measurements. The other kind of samples that we are getting is the hierarchical system, which is very important. So hierarchical samples, which means that they have the structural ordering when it's go to the higher length scale. The very good example is the plant material. So we have the molecules, we have the organelles, then it comes to the cells, then cells makes organs, then tissues, and the organisms. So the sense is a very nice technique because we can capture the structure of the samples in different length scale at the same time. So the example for these kind of samples are plants, cell wall, biopolymeric hydrogels, cellulose composite, or sedimentary rocks. The other type of samples are colloidal samples, which means like with the time, the sample concentration in the solution is going to be decreased, change because of the sedimentation. There's some examples are cellulose nanocrystals or soil nanoparticles. So how we going to use the cells? One is very important. So we are going to use the titanium cells to measure to fill the samples to make them ready for the beam line. The reason is the titanium cells are very easy to assemble and very easy to clean. So these kind of cells are very good for the large solid species, solid samples, various solvent like slurries, various plant systems, or biopolymeric hydrogels. We can change the path length of the cells by changing the spacer we are going to put it in. In the next slide, I'm showing you how we are going to assemble the cells. So we put first the O-ring. We have two different O-rings we can use in for different chemical environment. Like for water, we can use regular O-rings, but if you have like organic solvent, you have to use some special O-ring which is available with us. So we put the O-rings and then we put the first window in. Then we put the spacer, which is determined our path length. Then we put the samples aligned to the preferred direction of the beam. Then we put our second glass window, and then we put the again a ring to seal it. Then we screw the sample window to O-ring on the tight, and then we finally fill the samples, the solvent in. So it's easy, right? But the real challenge is not like that. So we are getting the samples like this from users. We are getting the plant stems. So we have to cut them into the uniform thickness to pack into the cells. So we pack them and then we put it. The real challenge that the one experimental challenge that have to face when we screw in it, even if we align them in the preferred direction, they flip. So it's a very big challenge. So it's sometimes we can manage it by when the sample size is still inside the cell. Sometimes we have to reassemble the cells. And the second challenge is that filling the solvent. We are trapping air bubbles. I am showing here, not like this is the purposely. If you can see, there is air bubbles. This kind of air bubbles you can get rid of maybe by simply tapping the cells, but sometimes it is also not easy to get rid of by simply tapping. In that case, we have to take all the solvent out, refill it. If that does not work, we have to reassemble the cell. So this is another example where we measure the sedimentary rocks in the deep sea, where these kind of rocks are important to do the methane oxidation under the anaerobic conditions. So this is the sample we are getting from the users. We have to cut them into the thin disc and then flip them in. So here I'm showing here is how we cut them and how we pack them into the cells. And sometimes even if we can use machines to cut, like to cut them to thin disc, but it's very hard to go to very thin slices by using machines, especially in this case. To avoid the multiple scattering, we have to go up to 0.3 millimeters. So in that case, I had to use my hands to do the sandpaper or the file. So in this experiment, we could capture the signals coming from the biomass associated with this kind of rocks and the size of the pores in the carbonate. Another example I'm showing here is the effect of expansion protein in hardwood pulps. The real life challenge of this kind of sample is when we fill these are like cotton fibers, so they are really get the air bubble traps. So what I had to do instead of putting the samples for injecting the solvent through the openings of the cells, I had to first pack fill the samples, then put some drops of water on the sides of the second window and flip it over and tighten the cells. So these slides, I remember Luke talked about it. This is how this is about measuring the sedimentary samples. I remember Luke talked about it, so I'm not going to talk about too much. So in that case, the sample is going to be sediment, so we had to use the tumbler to make them to have a uniform distribution across the time of the measurements. So in the very last slide, I'm going to talk what is the difference between the scattering patterns of the isotopic samples, like dispersed samples, and these hierarchical systems. As you see in the 2D image, now the scattering signal is symmetric across the detector. So what we do, we do the radial averaging. We basically define rings and we count all the neutrons comes within that ring, and we draw the points for the particular cube. But because of we have long structures in the hierarchical systems, we cannot, like as example in this case, the cellulose fibers, we cannot see the length of the cellulose fibers. So we have these asymmetric scattering patterns. So in this case, we do the wedge reduction. So this, we define two wedges, wedge 0 and wedge 1, and we reduce those two wedges separately. I'm sure Vinky is going to talk today or tomorrow in details about it. So when we have the wedge 1 completely coming from the isotopic part of the samples, wedge 0 is coming from the aligned features and the matrix of the system. So when we subtract wedge 1 from wedge 0, we can completely get the scattering coming from the aligned system. With that, thank you very much. If you have any question, we have 1 minute, as I promised you.
Thanks so much, Manju. Are there any questions? Salika.
Hi, nice talk, Wellington and Manju. I would like to ask if I get correctly, when you have a sample that is sediment quickly, then how it will be affect to the output, and what are the precautions you are taking, or it is the sample rotating or something?
Um, yes, it's exactly, you are correct. The tumbler is it's rotating. It's rotating with the cell, so it's always keeping the samples homogeneous during the time that you the measuring. That is why we did the tumbler setup for that one.
Okay, thank you. I have one more question if I have time.
Yes, go ahead.
Um, um, uh, when I couldn't remember the exact name, if I pronounce correctly, Walker talk, when we doing the contrast matching, contrast variation, uh, he described as you can do the contrast variation by using staining and using isotopes. So far I got to know how we can do the contrast matching using the isotope. So how you are doing it by doing staining?
I can answer that. I can answer that one. Sorry, that was maybe I went over this a little fast. I was comparing at the time microscopy and scattering, and in microscopy you use staining, and in scattering in neutron scattering you use isotopes. That's what I was referring to.
Okay, thank you so much.
We have another question from Jacob.
Yeah, I was curious if you've noticed a pattern with the presence of the His tag causing aggregation, because I'm sure Wellington, you know that that's a problem that we've been working with over here. And I'm wondering if you've noticed the presence of that his tag being a cause of aggregation regularly, or if it was just with that one specific protein system that you referenced.
In that, for me that I saw, actually was the first one that I was facing that issue. That clear was the his tag one, but I think I'm not sure if Hewitt's here, if he saw in the past some issues like that. We can do a
Do you-
follow-up with you for sure, but could it be the case of the his tag causing the
Sure, thanks.
segregation?
I think the question is how common is it that the his tag causes aggregation?
Yes.
Yes.
So I don't know. Kevin might have some input on this as well, because he has worked with a lot of proteins. I think he's still in the line. Kevin, do you want to say something about that one?
I don't really have much to add. It's kind of system dependent, and you can tweak things with buffers a lot of times to help minimize aggregation. And the same thing could be true with a his tag. So it all depends on the system and then the pH and the solution that you're using and how concentrated it is as well.
Sure. Okay. Thanks.
There's still one hand up. I think that's
Yeah.
probably still the same, isn't it?
It's true, Jacob. Yeah. Do you have any more? I don't see any more.
I thought I'd add one more comment to the practical aspects, practical hints. Something that sometimes one finds out the hard way. So don't find it out the hard way. Learn it from us. D2O freezes at about 4 degrees Celsius. H2O freezes at 0 Celsius, as we all know. So and common refrigerators often operate at around 4 Celsius. So it can happen if your refrigerator temperature is set low, you put a D2O sample in the fridge and it freezes, and that can destroy your sample. So just be aware of that. It's not very common that happens because a little bit of buffer salt and so decreases the freezing temperature again, but just be aware of that. It has happened to some of us. Thomas raised his hand, I think.
This is a follow-up to Jacob's, I think his name was, question. I have seen protein interactions with his tags are more frequent if the his tag is more difficult to cut off, presumably because the his tag lays back on the monomer, but may also form intermolecular interactions. So if you're having difficulty cleaving off your his tag, well, hopefully you design your his tag to be cleavable. But if your cleavage site, for whatever reason, doesn't work easily with your preferred protease, it may be that could also cause protein interactions and cause aggregation. So that's my experience, that they're correlated.
Thanks.
There was a question about hierarchical systems in the chat, and Venki answered it in the chat quite long, but I think it's probably of interest to everybody in terms of how that's defined, hierarchical system. We are using that kind of loosely. It basically means that you have different structural levels of organization, which is very common in biology, as everything builds from molecules to larger structures. So those are the kind of hierarchies we are talking about. But from the small angle scattering point of view, the significant thing about hierarchical is that it occurs at different length scales. So you have something at the few nanometer scale, and then you have something at the tens of nanometer scale, and something at the hundreds of nanometer scales. We often think in these kind of decades of organization, but that's just a hand waving way to put it. So don't take that too precisely as specific length scales.